Hello, welcome to Zigmatech Learning Hub. I'll be your instructor for biology. For this class, we are going to be taking our exercises from the Examguide app. Now, if you don't already have this installed in your device, I would like you to download the app in order to follow along in this class. Now, Examguide is a leading educational app that helps students prepare adequately for various exams. Exams such as the UTME, the post-UTME, WAEC, GCE, IJMB, KCPE, JUPEB, Calbepedia. In the junior sections, we also have the BECE, we have the SSCE, and so much more. Now, you can download the app from www.examguide.com or you visit the Google Play Store to download. Now, please subscribe to our channel and turn on the notification bell to update yourselves on new videos that will be coming up. Now, if you're ready for this class, let's get started. Today, we're going to be looking at Basic Ecological Concept 2. Basic Ecological Concept 2, we have actually discussed Basic Ecological Concept 1. And in Basic Ecological Concept 1, we actually looked at the definition of ecology. We also talked about some of the branches, the two main branches of ecology. We went ahead to look at some basic concepts that are used in ecology. And then we talked about local biotic communities in Nigeria and also local biotic communities or biomes in the world. So today we're actually going to be looking at some other aspects that relates or that is part of basic ecological concepts, or you can call it environmental biology. Now, in this particular topic, we're going to be looking at ecological factors. We're also going to be discussing population studies, and then we're also going to be looking at water holding capacity of different soil types. These are the three main subtopics we're going to be looking at. But at the end of this particular class, you are expected to note some things, and they are as follows. You should be able to define what ecological factors are, and you should also be able to define biotic and abiotic factors. You should also, by the end of this class, be able to mention some instruments used in measuring ecological factors. You should also be able to define what population is, and also some of the terms like population density, population size, population dominance. You should be able to define these terms used in population. And you should also be able to outline certain factors that affects population in a given area. And number 6, you should be able to describe sandy soil, clay soil, and loamy soil. And then finally, you should be able to define what we call water holding capacity of different soil types, and also be able to mention the factors that affects water holding capacity of a soil. All right. Now, these are the areas you're expected to know, and I've told you the subtopics we're going to be looking at. Now, let's start with the first one, which has to do with ecological factors. So today we're going to be discussing ecological factors under basic ecological concept. Now, what are ecological factors? By definition, ecological factors are simply those factors that influence living organisms and also cause a change in the habitat. They influence living organisms in a particular habitat, and as well, they cause changes in those habitats. And there are several factors we're going to be looking at, but basically they are divided into two factors. These ecological factors are divided into two basic factors. The first one is called biotic factors. The second is called abiotic factors. I repeat it again, biotic factors and abiotic factors. So we will be looking at what we mean by biotic factors. The name bio is gotten from a Greek word which simply means living, okay? So when we're looking at biotic factors, we are looking at those living factors, all living factors that influence other living organisms as well as their habitat. They are factors or living factors. Please take note, they are living factors. They are not just factors, but living factors that influence other living organisms and as well influence the habitat they find themselves. Now, examples of biotic factors include, we have competition, we have parasitism. Competition has to do with an interaction between two organisms where they struggle for all the necessities of life. We also have parasitism. It is also a type of association or biological association in which one organism benefits and the other one does not benefit but is harmed in the process. We also have another example called predation, and is a type of biological association in which one organism is killed for food, okay, while the other organism benefits from the other organism as food. The one that benefits is called a predator. The one that does not benefit but is killed in this association is called a prey. We also have mortality. We also have natality, okay? We also have food. We have diseases. We have pests. We have pathogens, we have commercialism, and we have migration. These are certain living factors. If you look at all these words, all these examples, you'll find out that it involves living organism. It is either an interaction or an association or a condition that involves living organisms. And that's why we call them biotic factors. The next is abiotic factors. Abio. Just the same way we defined a biotic as a living factor, abiotic factors are those non-living factors. And these non-living factors, they influence living organisms, other living organisms, as well influence their environment. They can cause changes in their environment. Now, examples of these abiotic factors include things like climatic factor. And when we're talking about the climatic factors, we're looking at things like temperature, we're looking at wind, we're looking at sunshine, we're looking at humidity, we're looking at rainfall, and so many other factors that includes climatic factors. We also have examples, another example of abiotic factor, which we can call chemical factor. Now, when we're looking at chemical factors, we're actually talking about mineral elements. And these mineral elements can include things like nitrogen, we have carbon dioxide, we have oxygen, we have water, and so on. We also have another example of an abiotic factor, which we call edaphic factors. Now, anytime you hear the word edaphic, it simply means it is relating to the soil. So edaphic factors are factors that relate to the soil. Now, they include things like the soil and its composition. They include things like soil profile, soil texture, and so many other things, the soil structure. These are what we refer to as edaphic factors. And then we also have another example of an abiotic factor, which is called topographic factor. Now, the topographic factors include things like the hills, we have the valleys, we have the mountains, and so on. These are topographic factors. Now, moving ahead, let's take a look at some ecological factors that is common to terrestrial habitats. We're going to be looking at the ones common to terrestrial habitats, and then we're also going to look at the ones connected or common to aquatic habitat. Remember, when we are talking about terrestrial habitat, we are talking about habitat that is occupied by land, or habitat, or you can call it the land habitat, okay? So it is a land habitat. Now, they include some of the factors that are common in terrestrial habitat includes temperature, we have rainfall, we have wind, we have light, we have pressure, we have hydrogen ion concentration, which you can otherwise call a pH. We have relative humidity. We have the edaphic factors we have just looked at. We have also topographic factors. These are some of the ecological factors you can find in a terrestrial habitat. Then we also have some ecological factors you can see or find or discover in aquatic habitat. Now, these ecological factors include temperature, yes. We also have rainfall. We also have wind. We also have light, yes, because we also have to talk about turbidity, okay? Now, when we're looking at light, turbidity, they move hand in hand, where turbidity actually talks about the degree of transparency of a solution, degree of transparency of a solution. We also have other factors like pressure. We also have pH. We also have salinity, which is the degree of saltiness of a solution. We also have dissolved gases. We have density, current, tidal movement or waves, and so on. These are some of the ecological factors that is common to aquatic habitat. And then let's talk about some instruments that are used for measuring ecological factors. There are some instruments, not all things you use, but we have specific instruments that are necessary for measuring ecological factors. In fact, they help you in carrying out several population studies, carry out several researches in the ecosystem, all right? Now, we have the first one. I'm going to just mention very few of them, but we have so many. We have so many, but very few. We have rain gauge. Rain gauge, as you can see on the screen, that is a rain gauge. And what is the purpose or the function of a rain gauge? It is simply used for measuring the amount of rainfall in a given area or habitat. It is used to measure the amount of rainfall in a given area or habitat. We have the other one, which is called a potter. A potter, you can see a potter. It has two suction ends, and then it's just a very simple diagram. It has two suction ends. You can also construct that at home. Now, it is used for catching small insects in farmlands and in fields. It is used for catching small insects in farmlands and in fields. You can see that you have two ends. One of the ends is where the animal is sucked in, and the other end is where you suck in as to get the animal trapped in the container. And you can also see a gauze, a wire gauze, helping to filter dirt from entering into your mouth while you are sucking the potter. Okay, so it is used for catching small insects like spiders, little spiders, and other insects in farmlands and fields. And then the next one is wind vane. Wind vane. Now you can see it, a wind vane. It also points to directions. You can see the north, the south, the east, and the west. That simply tells you it is used for measuring the direction of the wind. It tells you whether the wind is actually coming from the north, or if it is coming from the south, or if it is coming from the east or the west. And also tells you the direction it is going, all right? So it is used for measuring the direction of the wind. We also have the next one, which is called thermometer. Thermometer. It is actually used to measure temperature of a given area. It is used to measure temperature of a given area. We have so many types of thermometer. We have the maximum and minimum thermometer and so on. And but the one that is basically useful in terms of an ecosystem, not for the body, is the maximum and minimum thermometer. The one used mostly for the body is referred to as a clinical thermometer. Now we move to the next one, next instrument used for measuring ecological factors. We have the anemometer. Anemometer. Now, anemometer, it is used to measure the speed of wind. It is used to measure the speed of wind. Then we also have barometer. Barometer is actually used to measure pressure. And then we have the hygrometer. We have the hygrometer, which is used for measuring relative humidity. Hygrometer is used for measuring relative humidity. Please take note of this. It is used for measuring relative humidity. I have seen in several JAMB or WAEC questions where they asked another question. They said hydrometer. Please note there is a difference between hydrometer and hygrometer. All right. Now, hydrometer actually is used to measure density or relative density, while hygrometer is used for measuring relative humidity. Please take note of the difference. Then we have the next one, which is called the Secchi disc. The Secchi disc. Now, the Secchi disc is actually used for measuring the turbidity of water. Used for measuring the turbidity of water. And I told you that turbidity simply means the degree of transparency or clearness of that particular water or solution. And then we also have the measuring tape. And the measuring tape is used to measure the height and the length of objects. And then we have the quadrat. The quadrat, mostly used in population studies. It is used for population studies, mostly in terrestrial habitat, okay? Mostly in terrestrial habitat. And that is all you need to know for now about ecological factors. Now, the next thing to discuss under this particular topic is population studies. Population studies. Now, what is population in the first place? It is important to understand the definition of a thing. Now, population can simply be defined as the total number of organisms of the same species. Please take note of these keywords. Total number of organisms of the same species living together in a given area at a particular time. I repeat, it is the total number of organisms of the same species living together in the same place, living together in a given area at a particular time. Please also take note that every community in an ecosystem is made up of populations of different species. We have population of different species. You can find the human population, you can find the insect population, you can find the plant population. We have populations of different species. Are you getting me? So please take note, put that at the back of your mind. Every community in an ecosystem is made up of a population of different species. Now we have the next one, with what we're going to be looking at. There are some key words we will be talking about in an ecosystem. One of them is called population size. One of them is population size. Now, what is population size? It is the total number of individuals of the same species, total number of individuals of the same species in a particular habitat. In a particular habitat. For instance, we can take the number of lions in a valley, the number of lions in a particular habitat. Or you can say the number of buffaloes in a particular habitat. So the number of buffaloes talks about their population size. Just that. Now, when population size is large, the individual generally have a good chance of survival, which is true. It has a good chance of survival if faced with adverse environmental conditions. There is always a good chance of survival. Now, also in population studies, we have several calculations to consider. There are several times WAEC have brought out questions, and even also in JAMB, they bring calculations in biology. So don't be surprised. There are calculations to do in biology. Now, in this particular calculation, we're going to be learning how to calculate population density, and then we're also learning how to calculate population size. But for population size, the formula for calculating population size is population density multiplied by area of habitat. Population density multiplied by the area of the habitat. The next one is population dominance. Population dominance. Now, population dominance is when a few species exert a bigger influence on the habitat. Few species exert bigger influence on the habitat. Now, you find that mostly when you have humans or man in a particular community. The man becomes the bigger influence, becomes the populated dominance. They become populatedly dominant in that particular habitat, okay? So it is when a few species or when few species exert a bigger influence on the habitat. Now, when this happens, we say that they dominate and they tend to control and influence the environment around them. They dominate and tend to control. Now, one of the signs that they have dominated that particular place is the control and influence they have over activities in that particular environment. And then we move to the next one, which is called population density. Population density. Now, population density is the average number of individuals of the same species that can be found at a particular area of the habitat. Average number of individuals of a species that can be found in a particular area of the habitat. Like I told you when we're looking at population size, I said there are calculations involved. Now, there is a formula for calculating population density. Remember, we said that population size can be calculated as population density multiplied by area of habitat. But population density can be calculated using the following formula. Population density is equal to population size all over area of habitat. That is, if the population size is given and you want to calculate population density, it now becomes population size divided by area of habitat. Another way also of calculating population density is to calculate the average frequency, okay, average frequency of organisms over the area of quadrat, the area of the quadrat. Remember, we have just looked at the quadrat. You've seen the diagram or the picture of what a quadrat actually looks like, okay? So the average average frequency of organisms in that habitat, all divided by the area of the quadrat involved. And then finally, how do we calculate the average frequency? The average frequency is calculated as number of times an organism occurs divided by the total number of throws that are made. Total number of throws that are made, okay? Just put this in your back of your mind. We might see one or two questions that will involve calculations. Now, methods of population studies. Now, how do we carry out population studies? How do we carry out population studies in an ecosystem? We have two ways or two methods for carrying out population studies. Number one is by sampling. And that's by sampling, we make use of the instrument called a quadrat. We use a quadrat to calculate, or we use a quadrat to determine population studies in an ecosystem or in a particular habitat. Number two is by transect method. Now, the transect method, we use a tape or a measuring tape to carry out that type of population studies. Next is factors affecting population. There are several factors that affect population, and we're going to be looking at some of them. One of them is called natality. Natality. We also have mortality. Natality has to do with birth rate. Mortality has to do with death rate. Then we have immigration, that is movement of organisms into an area, and then emigration means movement of organisms out from a particular habitat. We also have availability of food. We also have natural disaster. We have diseases and so on. These are some of the factors that affects population or population size in a given habitat. And then we're also going to be looking at soil types. So that brought us to an end on population studies. Next is soil types. Soil types. We're also going to be looking in this particular topic, we're going to be talking about definition of soil and then types of soil. Soil types. Now, what is soil? Soil is the uppermost layer of the earth, which provides support and nutrients for plants and habitat for some animals. I repeat, it is the uppermost layer of the earth, which provides support and nutrients for plants, and it provides habitat for some animals. Now, we have different types of soil. We have two, for now, in terms of related to this topic, in terms of biology, we have 3 types. I know in agric they might want to include the 4th one, which has to do with silt, but for now we have 3 types. We have what we call sandy soil, we have clay soil, we have loamy soil. I repeat, sandy soil, clay soil, loamy soil. Now, what are sandy soils? Now, sandy soils are soils that are made up of high proportion of sand particles. They have high proportion of sand particles. And in fact, the particles in sandy soil are large and they are loosely held together. It means that they have pore spaces. That is, the particles are porous. All right. Another thing again you will notice in sandy soil is that there is it is actually low in nutrient as a result of high leaching. Because once water washes away the sandy soil, passes through the sandy soil, it carries a lot of nutrients away, and that is leaching. Another thing again about sandy soil is that it supports scanty vegetation. It supports scanty vegetation. The next type of soil is called clay soil. Clay soil. Now, clay soil is made up of high proportion of clay. High proportion of clay. It also have heavy soil because it is hard to cultivate. Very hard to cultivate, mostly when it is dry from being wet. All right. Then number 3 thing to take note about clay soil is that it has little ability to retain plant nutrient. It has little ability to retain plant nutrient. Another thing again about clay soil is that it can support light vegetation. And you see it in areas like savanna, okay, in savanna areas. We've talked about savanna in basic ecological concept 1. All right, next is loamy soil. Loamy soil. Now, loamy soil is actually the best type of soil for farming or planting. Now, it is made up of a mixture of sand and clay particles with high proportion of humus. Another name for humus is organic matter. High proportion of organic matter. Now, loamy soil, like I said, is the best soil for growing plants. Best soil for growing plants. Now, what is water holding capacity? What is water holding capacity? It simply means that what water holding capacity actually talks about the ability of a soil to retain water. You should be able to understand that water is very necessary for plant growth. Very necessary. It's also very necessary for photosynthesis. Very, very necessary. But the ability of plants to hold water determines how effective that particular soil is in terms of farming. All right. So water holding capacity of a soil is simply the ability of that soil to retain water. Now, there are several experiments that have been done on this, and it has been concluded that clay soil has the highest water holding capacity, and then sandy soil has the lowest or least water holding capacity. And there are reasons for this. Now, clay soil is regarded as the soil that has the highest water holding capacity because it's the particles of clay soil are fine and they are tightly packed together. They are tightly packed together. They are not loosed. Tightly packed together, and they have fine particles. And this makes it difficult for water to pass through them. So the rate of porosity in clay soil is very low. But in sandy soil don't hold water. They have the least water holding capacity because the particles are large, and not just large, they are coarse. So they have coarse particles and large particles, and these particles are not closely packed together, they are loosed. So porosity in sandy soil is very high, very high. All right, now let's take a look at some experiments to determine the water holding capacity of these 3 different soil types. Like I said before, whenever you are carrying out an experiment, please ensure your records are correct. You start with the aim, and the aim is actually your objective, what you want to achieve at the end of the experiment. Then after the aim, we have the apparatus. Now the apparatus include the materials you're working with. Then we also have the methods, which includes the step-to-step or step-by-step activities that are involved when carrying out the experiment. And then you have your observation. The observation is what you see or what you discovered when carrying out those procedures or methods. And then you have your conclusion, which is the final step to your records. And please take note, your conclusion must be the same with your aim. Now let's move ahead to look at how to determine the water holding capacity of these three different soil types. Now, as you can see on the screen, we have soil A, B, and then C. Now, you'll see that in each of the soil in the funnel, we used a cotton to actually block the mouth of the funnel so that the soil will not pass through it. Then we poured in the soil. Now, after putting in the soil, you mount the soil on a measuring cylinder, each of them. And then you pour equal amount. Please take note, it must also be equal amount of soil in each of the stands. Okay? For instance, in A, if we are using, let's say 50 grams, I'm giving an example, 100 grams of soil for A, we must use the same 100 grams of soil for B and then C. A stands for probably sandy soil, B is loamy soil, and then C is clay soil. That's where I'm looking at it now. And then when you are done with this, the setup, then add equal amount of water in each of the setups. For instance, we can add 100 ml of water in each of the setups. Then you allow it to drain, probably for 10 minutes. Now, after 10 minutes, you will discover that the soil, the setup that is A, setup A, okay? And setup A will have inside of the measuring cylinder, you will discover the amount of water in it has risen high. There is high amount of water in the measuring cylinder, okay? That's to tell you that the soil found in setup A did not hold the water, did not hold much of the water. So the soil A was so porous that water particles or water molecules should pass through it. Then if you look at B, there is moderate amount of water. There is still water in the soil, but also a good amount of water also has left the soil. And then when you look at C, you will discover that the amount of water drained from C is not much. It's actually small compared to A, B, and C. So in other words, in conclusion, C holds more water or retains more water, and C, like we said in the first place, is clay soil. B is loamy soil, and then A is sandy soil. So this is how we carry out an experiment to determine the water holding capacity of these three soil types. Now quickly, let's look at factors that affects the water holding capacity of this soil. First of all is the soil texture. The soil texture. Now the finer the soil, okay, because the texture of the soil talks about the degree of fineness of the soil structure. Now the finer the particles of the soil, the finer the particles of the soil, the higher the water holding capacity of that soil. While the coarseness of the soil, the lesser the water holding capacity of that soil. And then also number 2 is the clay content. If the clay content in a particular soil is high, the water holding capacity will also be what? High. Organic matter content too is also another factor affecting water holding capacity. If the organic matter content is also very high, it also causes the water holding capacity of that particular soil to be what? High. And then number 3 is mineral salt and pH, and then we also have the size of particles. Now this brings us to the end of this particular class. But before we go, let's try our hands on some questions from our Examguide app. We're looking at few questions here. Look at this first question. The activity of an organism which affects the survival of another organism in the same habitat constitutes A. Is it an abiotic factor? B. Is it an edaphic factor? C. Is it a physiographic factor? And D. Is it a biotic factor? Now they said there in the question, the activity of an organism. And remember I told you that when you're talking about living organisms, you're talking about biotic factors. So the correct answer to this question is D, a biotic factor. Next question, the instrument used for measuring relative density. I made mention of this. I said please take note of the instrument used for measuring relative density and the instrument used for measuring relative humidity. They are different things. The one used for measuring relative density is B, which is hygrometer. Hygrometer is used for measuring relative humidity. Please take note. Barometer is used for measuring pressure, and then wind vane is used for measuring the direction of the wind. All right. Next is which of the following instrument is used to measure relative humidity? Okay. Now you can see that the correct answer here is A, hygrometer. Now, which of the following is also used to measure the rate of water uptake in plants? Rate of water uptake in plants, and that is also one of the instruments, and it is called potometer. Option A is the correct answer. I want us to look at questions that involves a calculation. Now look at this. We've just looked at calculations that involve population studies. I'll use this as my last question we're going to be looking at considering. Now, in a field experiment, the frequency of water, remember I told you that is a way we calculate population density, all right? We said it is the frequency of the organisms all divided by the number of tosses. That's how we get the average frequency. And it is the average frequency divided by the area of the quadrat that gives us our final answer, okay? Now we have here the frequency of water leaf was observed to be 48. Okay, that's the number of organisms we have, how many they occur, and then after 20 tosses, okay, are of a 2 meter square quadrat, what is the density of the field? So what I'm going to do first of all, for me to find the average number of water leaf in that particular field is to divide 48 divided by 20. Now, using the calculator there, we have 48 divided by 20. Now, what we got there is 2.4. Now, 2.4 is not your answer because what they said is for us to look for the population density. And population density is simply the average number of organisms that occur divided by the area of the quadrat. And the average number of organisms we have just discovered is 2.4. Then the area of the quadrat. So what I'm going to do next is saying 2.4 divided by 2. And 2.4 divided by 2, my correct answer is D, which is 1.2 per meters square. 1.2 per meter square. Thank you for participating in today's class. You can practice more questions using your Examguide app. The app scores and gives a detailed explanation of all the questions at the end of your practice test. You can also learn particular topics of interest with different modes like study mode, mock mode, and even practice mode. It also has other features that makes learning very fun. Now, it is a must for all serious students. Download from www.examguide.com if you don't have it yet. See you in the next class. Don't forget to subscribe to our channels, hit the notification bell, and share the videos to your loved ones and friends that will benefit from it. Bye for now.